Hi, my name is Indira Sawh. My partner is Geselle Cancino-Prado, and our mentor is Dr. Kelley Healey. And our research was on the investigation of Candida auris Triazole resistance mechanisms and bioinformatic analyses to identify relevant genes for future research. Our summer research focused on the fungal pathogen Candida auris. We conducted a literature review of triazole drug mechanisms and performed a bioinformatic analysis to identify genes relevant to these resistance mechanisms. C. auris, like other Candida species, primarily infects and causes disease in immunocompromised patients. C. auris is an emerging fungal pathogen that was first described in 2009 and has caused outbreaks in healthcare facilities due to its ability to be transferred from person to person and sustain on surfaces. C. auris is divided into 5 clades based on genomic and geographical origin differences. There are currently 6 clades, including South Asia, East Asia, South Africa, South America, and Iran. C. auris has displayed high levels of antifungal drug resistance, including triazole resistance. Azoles act by disrupting fungal ergosterol biosynthesis by targeting the enzyme erg11. Ergosterol is similar to mammalian cholesterol. Because C. auris is an emerging fungal pathogen, research on its resistance mechanisms is limited. Through our literature review, several papers compared C. auris to another fungal pathogen, Candida albicans, because of their phenotypic similarities. From this, we hypothesized that C. auris utilizes similar triazole mechanisms of resistance as C. albicans. In our literature research, we analyzed 3 resistance mechanisms that have been studied in both fungal species. This includes point mutations found in the erg11 gene, which encodes for the drug target, overexpression of the erg11 gene, and overexpression of multi-drug transporters or efflux pumps, including CDR1, CDR2, and MDR1. In C. albicans, the overexpression of erg11 can be caused by mutations in the transcription factor UPC2, and the overexpression of the efflux pumps can be caused by mutations in transcription factors such as TAC1 and MRR1. One study demonstrated that deletion of CDR1 alone was sufficient to abrogate triazole resistance in selective C. auris strains. In addition, a recent study performed whole genome sequencing of 300 C. auris strains that revealed 54% of the isolates were found to have mutations in TAC1B, which is a transcriptional activator of the CDR genes. MRR1 is a transcriptional regulator of another efflux pump, MDR1, but studies so far show no clear influence in triazole resistance.
Mutations in erg11 in various clades of C. auris have been described and linked to triazole resistance. Our lab has previously characterized some of these mutations. Over the summer, we confirmed sequence analyses of 2 previously identified erg11 mutations found in South African clade isolates. Before the lab shut down, these mutations were individually reverted back to wild type through site-directed mutagenesis in order to determine their role in triazole resistance. The 2 original amino acid alterations included alanine to valine at position 125 and phenylalanine to leucine at position 126. As shown in figure 1, we were able to confirm that each mutation was separately reverted back to wild type. These engineered strains are currently being tested in the lab for their triazole susceptibilities. This work will tell us if both or only one mutation is linked to decreased triazole susceptibility in the South African clade. As we discovered in our literature review, another primary mechanism of resistance is the overexpression of multi-drug transporters that can pump triazoles out of the fungal cell. Therefore, we performed BLAST and PFAM analyses to identify potential genes in C. auris that are associated with this mechanism. As proof of concept, we blasted the C. albicans TAC1 transcription factor, which revealed several homologs in C. auris, including the recently described TAC1B, as shown in Table 1. Table 2 displays the identified homologs of another C. albicans transcription factor involved in the regulation of the efflux pumps MRR1. 2 C. auris genes were identified in the BLAST of C. albicans multi-drug transporter CDR2. Apart from the BLAST analyses, we also performed PFAM analyses to identify commonly conserved regions and gene families among all cellular factors. Overall, the PFAM analyses revealed a commonly conserved zinc cluster family ZN2 cis6 binuclear cluster domain between the transcription factors TAC1 and MRR1, as shown in Table 4. The PFAM analyses also confirmed C. auris CDR1 and CDR2 homologs belonging to the ABC2 transmembrane families, as shown here. Based on all our findings, we hope to further our research by determining which ERG11 mutation in the South African isolates of C. auris lead to reduced triazole susceptibility, measuring gene expression of the homologous drug-efflux pumps within isolates of C. auris, and analyzing sequence of an homologous transcription factors in clinical isolates to identify and characterize additional mechanisms of triazole resistance in C. auris. Thank you for listening.